All right, everybody, we're ready for our next demo. Will be presented by Changjing Wang, who will be talking about igniting full-length isoform and mutation analysis of single-cell RNA-seq data with flames.
So if you want to follow along, you might just want to open up the workshop page and just search up flames and fire up that Docker image. One thing though is that I noticed that when you fire up from this service and you click this URL, at first you'll see some sort of error message. For some reason, for this workshop, it takes up exactly 5 minutes for that image to start working. So maybe just pull up a timer and remind you when that is actually ready. And for this first 5 minutes, I'll just go through some introductions before you can run the workshop.
Oh, you might begin with open.
Yeah, it'll keep saying some sort of.
I vote you that gateway.
Yeah. It'll be that for like the first 5 minutes, and then after that, it'll be fine.
There's a comment about the audio. If you can maybe move it closer.
Is there a podium mic? Are you wearing
I
it?
think my mic broke.
Oh, it's slid.
Yeah.
Put it on your shirt maybe instead.
Yeah. Okay, is that better?
That's
Mike says yes.
yeah.
So first off, so we heard a lot of talks about single-cell RNA-seq talks today and the days before. And one thing though is that with traditional 10x, say 10x and regular short-read sequencing, really you're only getting the first 100 or 200 bases from say the 3' end, and there's only barely enough to identify which gene is being transcribed, but we all know that it's actually the transcript that eventually gets turned into proteins, and a gene might have several transcripts. They may have very different functions. Some could be even having the opposite effect. So just only identifying which genes are being expressed is really not the full picture here. And apart from that, if we actually combine long-read sequencing with the single-cell kits, apart from getting the whole transcript, we can also pick up some, say, SNP or INDEL mutations along that gene. And that could be particularly helpful when you are analyzing samples from cancer data. For example, if you could identify certain SNP, certain cancer clones with a SNP, you could then compare the transcriptome profile of cancer cells with that SNP mutation versus, say, another clone that you're interested in, because this is being profiled simultaneously, both like the SNP mutation, gene expression, and transcriptome expression profile. And so there's been a lot of studies combining single cell with long-read sequencing. And our previous work published on genome biology is one of such, where we combine the 10x single cell kits with nanopore sequencing, basically because with nanopore sequencing, yes, we have longer reads, but the problem is the cost is much steeper, and you get lower coverage, lower depth. And so what we did is, instead of having long reads on the entire sample, we subsampled the gems, which is effectively subsampling the cells, and we performed long read sequencing on only the smaller subsample of the cells, so such that those cells will have a deeper sequencing saturation. But there's other ways of configuring this. For example, instead of subsampling the cells, maybe you're only interested in, say, like a panel of genes. So you could have more number of cells in your experiment, but you're only doing long-read sequencing on a panel of genes. And that's described in this other study we had with SC-RNA-seq. And apart from that, yeah, there's really a couple of other studies doing combining long reads with single-cell sequencing. This LR-SPIC is one of them, and this one is actually pretty interesting as well because they use combinatorial barcoding, which means that you don't actually need any microfluidics equipment at all with it. And I think
one question: are these FLTC protocols? Are those now commercially available or only in your lab?
no, it's only a couple lab, yeah.
So would this work with PacBio long reads?
I haven't tried. I haven't managed to get my hands on it.
Because they partner with 10x, and you can go from the Chromium into the make for the PacBio.
PacBio and 10x.
Yeah, so that's
collaborate on
commercially
something
available.
like this.
I think it's commercially available now.
Okay. Do you know how the platform's called by any chance?
I'm trying to see.
It would be interesting.
I thought PacBio had their like own single cells.
Yeah, they partnered with 10x to make a library kit that will make the libraries appropriate to sequencing on PacBio rather than Illumina.
Sounds good.
It's called Kinex.
Kinex.
Kinex, yes, Kinex.
Yeah, this one.
And actually, yeah, they had it first for single cell, and then they put it out for bulk. I think the Kinex kit.
Cool. I think it's over 5 minutes now, so if you try that link again, it should be working now.
Let's get like 5.
6 minutes.
I think the Docker image is like 2
So yeah, if that URL is working for you, maybe you could you prefer to follow along. That would be great. Just run this first chunk here.
Is there a link to the vignette or?
Oh, like the rendered one.
Oh,
I-
yeah.
I can put it in the Slack. I put it in the online.
Yes, please. Thank you.
Yeah.
Did it get updates?
Yeah, just pop that one in the. Right. So our Flames pipeline is designed to sort of help you process and analyze such data, especially with the 10x nanocore combined experiments. And so the first step will be demultiplexing the reads. And what do I mean by demultiplexing exactly? That's because with our raw reads, it will look like something like this, where you have the so this picture illustration below is directly from the 10x user manual of how their library will look like. We'll start with some sort of adapter, and that will be this thing here. It's the adapter, and then you'll have 16 base pair of cell barcode, and then 12 UMI, and then the poly-T sequence, and then the gene insert, and then followed by some other adapter on the other end. So the first thing we need to do is to locate where the barcode is and trim off these adapter sequence. And because we had a pair-coupled short-read sequencing, we know exactly which cell barcodes we're expecting. And given that information, we would compare the barcodes extracted from here to our allowed barcode list that we gained from the short-read sequencing. And of course, allowing, say, 1 or 2 edit distance just to make up for the slightly higher sequencing error with nanopore. So to do that, we actually have a very flexible way of handling this. You can specify your protocol by something like this, where you pop in the first adapter sequence that you're expecting. You could name it whatever you want. It just has to be in the same order as they appear in your protocol. So the first thing that will appear in our 10x protocol here is this adapter sequence, and then it's followed by 16 barcodes, nucleotides, because they could be anything. So we'll just have 16 ends, and then 12 ends for the UMI.
So you said the primer, the index, not the simple index, the
Barcode?
adapter
Yeah.
is required?
This sequence?
Yeah.
Yeah, we kind of need to know what's before and after the cell barcode.
Because I was just thinking, because like our sequencing center trims those off for us already.
Oh, you don't have anything before the barcode.
Yeah, no, they give us demultiplex data, and we
Oh.
get R1, R2, I1, and I2. And that's what we get as output. We don't get.
Oh, isn't that like short-read sequencing?
Oh, no, we get that also for 10x.
I mean, like short-read with 10x, right?
Oh, is this with the long one stuff?
Yeah, this is the long-read stuff. Because with
Okay.
short-read, there's like a lot of well-developed
Okay, this
stuff.
is the long read stuff for the PacBio. Oh no, not the PacBio.
Nanopore.
Nanopore.
Yeah, because with
Is that-
with the regular short read and the next one, you get like very good software support from the vendors like Cell Ranger and stuff. You don't really need to worry about this. Whereas if you do nanopore with 10x, it's a bit niche, so we need to process this ourselves. And yeah, we depending on how your protocol works, you just put down sequence in the same order as you expect them. And let me run that chunk here.
Sorry if I missed the intro in the beginning. So you're doing this to perform then the demultiplexing, right?
Yeah, this is for demultiplexing the 10x nanopore reads.
Yeah. So it's particular like for nanopore 10x.
Yeah. Well, if it's Illumina 10x, you just run Cell Ranger, right?
Yeah, I was thinking of like BD Rhapsody data, for example, like for single cell RNA-seq.
BD.
Rhapsody. It's like 10x. It's just another technology.
Oh, if it also have some sort of cell barcode, and then you have some sort of primer, you should be able to put down that pattern and specify it, and we should be able to demultiplex it. And let me run that chunk. And let's try demultiplexing. So let's just get a empty folder first, and we grab the barcodes that we expect from this file here, and we run find barcode to do demultiplexing. How's that look? Yeah.
Quick question. What does this assume about the positions of the UV and the barcode?
So the positions are in the same order as you specify them here.
So you hop around.
Yeah.
Can you put something in between the barcode and the UV?
Yeah, you could put something in between. The only thing that's required for this pattern here is the keyword barcode and UMI. Anything else, you can name it whatever you want. You can put it wherever you want, between UMI, after UMI. Yeah. And let's have a look at the raw read before we demultiplex it. Yeah, this is the first read from the raw FASTQ file, and let's have a look at the demultiplex one. Yeah, so we'll just find the barcode and UMI, and we trim it off, but we stuck it into the identifier field of the FASTQ file. So yeah, that's this field here. And after demultiplexing, next thing we do is, oops, sorry, I'm getting ahead of myself. If your experiment design is long reads only and you don't have short reads, so you wouldn't have that list of barcodes that you're expecting, this is also okay with our Flames pipeline. You just run this blaze command here, and you tell it roughly how many cells you're expecting, and you just give it the FASTQ files. And instead of trying to match barcodes found in your long reads with a whitelist or a allow list, it will just count the number of occurrence of those barcodes, and for those that are more frequent, it will think that it's most likely the correct cell barcodes, whereas the less frequent one, it will assume that it is actually some correct cell barcode with an additional sequencing error. So it will try to match those ones to the more likely cell barcodes. And yeah, you probably don't want to run this line on the Docker image, because I think it will take up a bit more memory than it's allowed on that platform, and it might just crash your session. But I'm running this on my own server, so it's fine. And after demultiplexing, next thing we do is we align the trimmed FASTQ reads to the reference genome. And we do that with Minimap2, and then we sort and index it with SAM tools. The caveat there is because we're calling these binaries outside of R, so you need to let Flames know where those binary lives. So preferably if they're found within the path variable, Flames will just use it. But I think the package manager on Ubuntu, when you install Minimap2, it doesn't come with the K8 binary for some reason. So you might want to just specify that as an additional parameter.
What genomes are available, or can you make your own, or?
Oh, you just provide your own. So yeah, you just have a FASTA file as the genome, and then you provide annotation as in GTF, GFF format.
All right, so it'll make the reference for you and everything, or the index.
Yeah.
Whatever.
Yeah.
Does it.
And yeah, if you're on the Bioconductor workshop image, don't actually run this chunk, because Minimap2 actually takes up quite a bit of memory, and it might crash your session. Sorry about that. But yeah, if you're on your own computer and you run it, it's basically what you would expect. It's no difference from running Minimap2 yourself. And
we have a question from the online audience, but I think it's clear, asking, does Flames work with short reads only? Sorry if I missed.
Oh yeah, if you only have short reads, then you probably wouldn't use Flames. You just use the regular cell ranger and then the other standard packages on Bioconductor. And following alignment to genome, we then run isoform identification, and then we realign it to the transcriptome, and then we do some quantification. So for the interest of time, we won't be running those steps side by side. And Flames actually provide a sclong pipeline function that runs all of those steps altogether for you. So let me see if I can get this working.
So how long does it take and how much memory does it need?
The step that takes up the most time and memory will be the alignment step. So whatever amount of memory that you need to have Minimap2 running, that's like the upper limit that Flames will actually be using. I think Minimap2 is multi-threaded. Yes, because Minimap2 is multi-threaded. You just provide a thread argument to Flames, and it will just pass that to Minimap2. Some of the other steps in Flames, I know that the identification and quantification part are single-threaded, but I don't think that takes any longer than the alignment step. So we'll probably work to have a multi-threaded version of that, but yeah, at this point it's only single-threaded. So after you're done with running this whole pipeline, you get a single-cell experiment object, and let's see. Yeah, so the counts will be the transcript counts. And then the isoform annotation will be also stored there in the, I think it's the row ranges slot. You can just save this as a GTF if you want, but otherwise it's in this single cell experiment object. So after we've run all this, we finally get this single cell experiment object, and we can start some actual analysis. So yeah, because it's a single cell experiment object, you could just do pre-processing with your favorite bioconductor packages, scRNA, Skater, right? And for this workshop, we'll be using the cell line mixture data from our previous genome biology paper, and that's saved in this file here. So if you run this chunk, it should load up that object. And let's see, SCE. Yeah, we have 160 cells that are coming from 5 different cell lines. So they will also all be having different SNPs. And this is the object that you will get at the end when you finish running Flames. I've also stuck a gene count matrix as a ALPS expression slot in the single cell experiment object that comes from running gene quantification from Flames. So it's gene counts from the long reads. Although we do have short reads, but I mean, why not? Just for the kicks, let's see how the gene counts from long reads work. So you could just run these standard pre-processing steps. You probably wouldn't want to run this chunk because for some reason they'll take a bit too long. Maybe I had too much cells in this demo data, but I had this object here, and it's the single cell experiment object, but with these PCA stuff run already. So if you just load that up, we can see these 5 different cell lines, pretty distinct on the UMAP, and they're being clustered as you'd expect. So let's start with some mutation analysis. So Flames provide the let's pretend that we don't know any information about the mutation in these cell lines, and we can use this find variance function to get a bulk count from the alignment file. If you ever use SAM tools, this is very similar to the SAM tools PALLAP command. it will just give you a little bit of extra information, including, let me just run this chunk real quick. It will just give you some additional information, such as the gene name from that region and this homopolymer %. And this is because with nanopore sequencing, it gets a bit confused when you're in a region where it's entirely just the same nucleotide repeated again and again, and nanopore would have trouble figuring out exactly how long that stretch is. So it'll have much higher sequencing error in those regions. So I just put this column here as a way such that if you see this percentage really high, you'll know that say 50% of that region is of one of the same nucleotide. That's 50% is actually okay, but if you see it's like 90% or 100%, then you know it's probably a nanopore specific sequencing error. You probably just want to check that out.
Yeah, another question. Could you kindly explain the log normalization step from the online audience?
Log normalization. This is just pre-processing from using the other Bioconductor package. It's not really relevant for this workshop. It's just so that I can run PCA and UMAP. So if you're interested in these steps, you might want to check out the Scuttle and Skater package. Oh, looks like it's thinking a bit hard there. And yeah, we've got this counts here, and let's filter these potential mutation loci by doing some simple tidyr command. So let's look for the ones that have a alternative allele frequency, not too high and not too low. And let me run this chunk. Looks like we're left with a couple. They all got a pretty large number of reads supporting them. That's good. And the frequency looks about right if we're dealing with cell line data. So after we run this, we can get these, because these counts are still at the bulk level, because we have the single cell data, so we can use the SC mutation function to call mutation at the single cell level. So that's this chunk here. This might take a while. So this function basically just takes the band file, and for loci that you specify with these, so this is the chromosome name and this is the position. For loci that you specify here, it will count the number of reads supporting each allele at the specific loci for each cell. And yeah, you also need to supply the cell barcodes here as an argument. And I think this one is multi-threaded. Like, let's just double check that. Let's run top. And yeah, I think this one's actually using multiple threads and should be pretty quick. Maybe I should just blow up this. Okay, it looks like this is taking a bit longer than I expect. Oops, finally done. Let's have a look at what we get from SC mutations. Yeah, so we get a table again, and this time we have the allele and the cell barcode, and then this count is the number of reads supporting this A allele at this position for this cell barcode. So now that we have the SNPs for each cell, let's overlay this on the previous UMAP and see how it looks. So let's go ahead and, yeah, because we get a table and we can just use some good old tidy R verbs to summarize this to finally give each cell a label to say whether it's mostly the A allele or the T allele at this position. So because this is the cell line data, I know that these cells are homozygous. So I just basically do, for each cell, I'll just call whatever allele when it's having over 50% of reads supporting it from that cell. So we'll run that and we'll plot it on the UMAP from before. So for this one, you can see it has the T allele on this one cell line, whereas for all the other cell line, it has the C allele. Let's run this whole chunk actually. And for these, I don't know, 5 different loci, we can see that they actually distinguish the 5 cell lines perfectly, I think. Yeah, this is a very clear cut because it's cell line data. If you are actually dealing with cancer data, it will get a lot messier because it might be 50-50, or I don't know, it's cancer. It might have more than 2 copies, right? So you might want to think more about that. And since we have the transcript count matrix, what we can do is we can do some differential transcript expression analysis. And for this purpose, let's just pretend that instead of cell lines, we have cancer data. Let's pretend that we have 2 samples, and we then identified 2 distinct clones using SNP. And yeah, for that, I'll just drop one of the cluster and pretend the other 2 are replicates from the 2 different cancer sample. Let's do an MDS plot. So yeah, we can see the 4 samples. There's a bit of distance between these 2 and these 2. So let's just pretend that these ones, 1 and 4, are cancer cells with one particular SNP, and they're coming from 2 different patients. And these are cancer cells with another SNP, and they're coming from the same 2 patients. And we can do some differential transcript expression analysis. Let's just go ahead with lemma because I find that to be pretty simple. Yeah, if we run this chunk, we get some differentially expressed. How am I doing with time? Right.
Actually, we got 8 more minutes.
Oh, okay. Right. Yeah, we get some differently expressed transcript, and let's see how that looks on our UMAP. Yeah, and you can see indeed that these 2 got slightly higher expression than the other 2. And another thing you can do with transcript expression is differential transcript usage analysis. And I got this really nice figure from this other paper that highlights the difference between differential transcript expression versus differential transcript usage. So the usage will be testing for the percentage of that transcript rather than the absolute expression level between the two conditions. So let's do that. I'm not sure this chunk is going to finish within time. But yeah, that's pretty much it anyway. So to test for differential transcript usage, we can basically just do some simple chi-square tests to test against a uniform distribution. And for that purpose, instead of using the transcript counts, we'll be aggregating transcript counts that have the same splice sites and treating them as if they're the same transcripts, because even with long reads, we still get a bit of degradation at the end, or sometimes nanopore could still not be like long enough. So if all the splice size matches and it's only differing by the first and last exons a little bit, maybe we'll just pretend that they're the same transcript for this purpose. And to get that done, we need to have this annotation file, which is from the Flames outputs, but it's just that we're not running the whole pipeline here. I'll just download that and pretend that we have run it, and it's in the output folder here. And if we run this, we get a data frame, and we have the gene ID here and adjusted p-value. Let's see if they actually have differential, if the transcript usage is actually different here. Oops. Can I blow this plot? I'll make this bigger. Image in still a bit washed out, but because for FLISSEQ we actually have a larger subsample with only the short read. So for this plot, I actually used UMAP that's using the short read sequencing for the whole, like both the smaller and larger subsample. And the ones that we don't have a transcript count, I'll just plot them in gray. So those will be the ones that are not selected in the smaller subsample. And for the cells that we do have a transcript expression level, we plot them, we color them by their expression level. And these are the 4 different isoforms coming from that gene. And you can see indeed they have seemingly a bit different transcript usage among these 5 clusters. And I think that's about it. Yeah, so if there's any questions.
So the object that you read in has got both short and long reads in it? And this is
The single cell experiment object that I read in is just the long read one. But for the last function, I actually grabbed the short read. Yeah, just to make this extra pretty figure. Yeah, if I just have 150 cells, it wouldn't look as great, right?
You got shut down. We're still.
Oh.
What? Why are the slides in here?
Oh, yeah. It's a bit wobbly.
Okay. All right, well, I guess if there are no other questions, I think we have half an hour break, and then there will be one last package demo, and then after that, I know we've got some final remarks, so. Getting down to the end of it, let's go see if there's any cookies left.